Hello, students. This is Dr. Faizan Mirza. Here today, I have taken the question regarding gene therapy and ethical reasons and using primers and disease called the sickle cell anemia. So let's start discussing this question. This is from October number 20, variant 42, the second variant. So I'll be just discussing this one question here in this video. So let's start. Sickle cell anemia is a non-infectious disease and a chronic illness. If not treated, sickle cell anemia can be painful and life-threatening. Further, we are telling sickle cell anemia is caused by a base substitution mutation. We know what is base substitution mutation, where a base gets incorporated in place of another base. So if there was a T in the DNA sequence, it gets replaced by another base in the gene coding for beta globin polypeptide of hemoglobin. This leads to changes in the primary structure. We know when the base changes, the codon changes, and when the codon changes, the primary structure changes may change because different amino acid gets placed. And when the codon changes in the amino acid change, we call that as a missense mutation because the missense mutation means that amino acid which was supposed to be there, it's not there, and another amino acid is actually in its place because the codon got changed. So here they are saying it's valine is present inside of gluten instead of glutamine. So yeah, this is the codon changing, and this is definitely a missense mutation. This is also an abnormal sickle shaped cells. Since the primary structure changes, so the hemoglobin will not be of the same shape. The beta globin chain will change. The cells stick together with the blood vessels. Symptoms of sickle cell anemia include painful attacks when nervous cells block capillaries in tissues and organs, suggest the consequences of cells when sickle-shaped red blood cells block capillaries in tissue and organs. Now, this is something that they expect you to recall from your AS, that when blood flow is affected, so how should this actually be difficult for a cell or a tissue? So these cells will be provided with insufficient oxygen to the tissues. We know RBCs provide oxygen to the tissues, so insufficient oxygen will be provided. These tissues cannot carry out aerobic respiration. You need to connect both. That oxygen is required for aerobic respiration. And since the oxygen will not be there and aerobic respiration not happening, so the cells may not function properly. Sickle cell anemia is an autosomal recessive inherited disorder. What is an autosomal recessive disorder? It is caused by a gene which are present on the autosomes. It is a recessive illness, which means the allele which is abnormal is a recessive mutation. So both chromosomes of the homologous pair must have the same recessive allele for the disorder to be there. If one homologous chromosome of the pair is having a dominant allele, the other is having a recessive allele, the person will be a carrier. So allele HbA codes for normal beta globin, HbS codes for sickle cell anemia. So if both HbA are present, the person is normal. If one Hb and HbS are there, the person is carrier. If the person is having both HbS, the person is sickle cell anemic. People who are heterozygous have sickle cell trait, sickle cell SCT. For a child to inherit SCT, sickle cell anemia, both HbS must be there. So the parents both must be carriers of the sickle cell trait. Only then there is 1 in 4 chance you can just perform the cross and see that if you cross both of these together, there will be 1 in 4 chance or 25% chance of the offspring getting the disease. A genetic screening program is available for sickle cell trait. When a mother is screened and found to have SCT, the father is then screened. So this is the screening of the parents, and the mother is being screened, the father is being screened. So this is before fertilization occurs. If the mother becomes pregnant, the fetus is screened for both sickle cell anemia and SCT. Now, in this case, the fetus is there. The test is done either by amniocentesis or chorionic villus sampling. We have studied both of which carry a small risk of pregnancy failing. Okay, outline two advantages of genetic screening for SCT and sickle cell anemia. So if the child does not have the mutation, it removes the worry regarding the disease. If the abnormal the allele is there, then the parents can be informed about the consequences and the challenges associated with the disease so that they are in a better state to take decision regarding the cost of treatment. So these you basically need to just outline two advantages here. So to test the presence for the presence of HbS, DNA is extracted and the polymerase chain reaction PCR is carried out with two specific primers. One mutation to produce HbS is a base substitution mutation in the sixth codon of beta globin gene. The normal codon GAG changes to GTG. So this is the point mutation. GAG changes to GTG, a point mutation, a missense mutation, or a base substitution mutation in which there is a change of one base, the second base of this codon. The normal specific primer detects GAG. whereas the mutant specific primer detects GTG. So two specific primers are there. One can detect the normal codon and the other detects the abnormal codon. Explain why primers are used in PCR. Okay. How the use of two specific primers allow amplification of the normal sickle cell anemia and SCD genotypes. What do the primers do? We know the primers attach to the 3' end of the single-stranded DNA, making that particular part double-stranded. Also, this if this is the DNA and this is 5' and 3' end, so the primer will attach along the end, which is 3 prime. This is the part where the primer attaches. So this part becomes double-stranded. When this part becomes double-stranded, then Taq polymerase can attach here and elongation can continue according to the direction of 5 prime to 3 prime. So coming back. Primers attach to 3' end of single-stranded DNA, making it double-stranded. They anneal to the DNA that is in turn allowing Taq polymerase to bind to it. It begins elongation. This allows DNA to be amplified. So we have explained how it's used in PCR. Furthermore, the type of primer binding allow identification of the genotype. If both normal primers bind, then it's HBA. HBA, we are told that the normal specific primer identify GAG, whereas the mutant specific primer identify GTG. So if both normal primers bind, then it's HBA. HBA, if both mutant primers bind, then HBS. HBS, if both primers attach, then it's SCT is confirmed. So all those three genotypes can be considered here. Furthermore, they are saying after PCR, gel electrophoresis is carried out on the products of PCRs. Figure 4.1 includes the results of two individuals A and B tested for sickle cell allele. Each lane is having 860 base pair band to indicate the test is valid. So that's just for the validity of the test that, okay, this is like a control, or you can say validity is confirmed. Lane one is a control G lane with 207 base pair for an individual of known normal phenotype. So this is lane one. You can see two bands are forming here. One for 860, one for 270. Both are there. Sorry, 207. Lane 2 is a control lane of 207 band for individual known with sickle cell anemia. So lane 2 again is with sickle cell anemia. So it's again showing 207 at 207. Lane 1, 3, and 5 contain DNA from PCR used for normal with that normal specific primer 1, 3, and 5. So we know that 1, 3, and 5 are normal. Then 2, 4, 6 contain PCR with the mutant specific primer. So 2, this 2 here, this 4 here, and the 6 here. So these contain the mutant ones. So how do we confirm this? We confirm this by the particular bands that form 1, 3, and 5 control PCR with the normal specific primer. So these are normal 1, 3, and 5. Whereas 2, 4, and 6, they are both the ones which have the mutant primers. So based on this, what we can see, individuals of lane A, individual in B, what do they have? Lane 3, they are having 4 bands here, 2 bands in lane 3 and 2 bands in lane 4. Whereas lane 5 of individual B is showing only the 860 base pair, nothing along the 207, whereas the lane 6 is showing both. So how do we interpret what genotype is individual B is having, what genotype is individual A, showing all the 4 bands here, whereas individual B is showing 3 bands here, like 1 in lane 5 and 2 in lane. So how do we interpret this? So did you use the genotype of the phenotypes individual A and individual A and B? So SCT is there, HbA, HbS, HbS, HbS is sickle cell anemia, the person A and B. So this is how we identify this. Now, what working we have done for this? So for that, we go back to the last two bullets, which are told to us, where we are told that the lane 1, 3, and 5, they contain normal specific primers. So if we see bands there, so that means the normal codon is present. So lane 5 is not showing the codon here. It means the normal codon is absent. So if the normal codon is absent, that means the person is lacking the normal allele, and the person must have both abnormal, the HbS codon. Whereas individual A is showing the bands for both the normal specific primer as well as the mutant primer. So all 4 bands are here. So that person must be a carrier or SCT trait. The number of cases of sickle cell anemia is highest in sub-Saharan Middle East India. In India, the areas that have high incidence of malaria suggest people with SCT heterozygotes are either unaffected or may have mild symptoms of sickle cell anemia, but advantage of SCT is increased resistance to malaria. Explain how natural selection operates and maintain the sickle cell alien population with malaria. So here you go to the selection and evolution. We know that SCT people have selective advantage. They survive. When they survive the selection, they survive both the selection pressures of malaria as well as SCT, as well as the sickle cell anemia. So both selection pressures they actually survive from, and then they basically reproduce. They pass on those alleles to the next generation. So that's an example of a stabilizing selection where we have this kind of a graph that continues there. These individuals are, for example, HbA. They are less in population. Individuals who are HbA, HbS are more in population. People who are HbS, HbS, they are less in population. So this is an example of stabilizing selection. Moving on, the last part. Parents who have used IVF to produce embryos may decide to have embryos genetically screened by a test known as pre-implantation genetic diagnosis or PGD. Only embryos that do not have sickle cell alleles are transferred to the woman's uterus. Discuss two ethical reasons why parents using IVF may choose not to have PGD. Now, knowing that PGD is actually advantageous, you get to know about the embryos, whether they have the disease or not. So that's pretty useful. But still, if the parents are not considering to have PGD, why? Because embryos not transferred will then be discarded. Now that's a basic ethical reason that what happens to embryos which are not used. This can be a cause of concern regarding religious, ethical boundaries of the person. Parents may be pushed to choose gender of the baby, which is inappropriate. So yeah, when the people know that PGD, okay, this is one gender and the other gender, they may end up choosing one gender over the other. So I hope this video was helpful for you. I would recommend that if you have not attempted this question so far, so go and attempt it yourself and try to just see if this video was helpful for you. That's it from my side. Thank you so much.